Welcome to ELISA 101.
Bro, this is immunofluorescence.
Welcome to Immunofluorescence 101. Immunofluorescence is a technique used to visualize the location of a protein in a tissue using an antibody with a fluorescent tag. The primary antibody binds directly to the protein of interest at one of its epitopes. An epitope is essentially an antibody binding site. A secondary antibody is designed to target a primary antibody in order to provide signal detection. The secondary antibody will contain a fluorescent tag, which will fluoresce in the presence of light of a specific wavelength. Although secondary antibodies are not essential, they are used to reduce cost and increase flexibility, as the same secondary antibody may be used to detect many different primary antibodies.
We will be talking about antibody selection, monoclonal versus polyclonal antibodies, and antibodies in immunofluorescence microscopy.
It is important that the primary antibody is produced by a host organism of a different species than the specimen to avoid cross-reactivity between the secondary antibody and the antibodies present in the specimen. If the primary antibody is from the same species as a specimen, this will result in the secondary antibody having affinity for both the primary antibody and the specimen. Secondary antibodies are made in a species different from both the primary antibody and the specimen in order to minimize nonspecific binding that leads to false positives and high background noise. Monoclonal antibodies target a single epitope within an antigen and are created using identical immune cells cloned from a single parent cell. In contrast, polyclonal antibodies are created using a number of different immune cells that have affinity for many epitopes within the same antigen. Polyclonal antibodies can be used to amplify a signal from the protein of interest with low expression levels due to their affinity for multiple epitopes within the protein. On the other hand, monoclonal antibodies are better for visualizing a signal from a protein of interest with moderate to high expression levels while minimizing background noise and cross-reactivity due to its high specificity. Protein targets can be visualized because of a fluorescent tag that is found on the secondary antibody. The gene for the secondary antibody is modified to encode for the fluorescent tag. A primary antibody binds to an epitope on the protein of interest, and the secondary antibody binds to the heavy chain of the primary antibody. This linkage results in the protein of interest being in close proximity to fluorescent tags, which, under a fluorescent microscope, results in the visualization of the protein. In summary, it is important to consider the species of origin when selecting antibodies to prevent nonspecific binding. Monoclonal antibodies target a single epitope within an antigen, and polyclonal antibodies target many epitopes within the same antigen. Antibodies are used in microscopy to visualize proteins by using a fluorescent tag bound to a secondary antibody.